Hello students, we are into a new chapter. Name of the chapter is Antibiotics. I know this term is very familiar to you. You have come across this. Let's study in detail about antibiotics in this chapter. You are learning antibiotic, as a pharmacy student here. Let's go to the definition first. Antibiotics, they are chemical compounds. They are chemical compounds derived from or produced by living organism, and their synthetic analogues, which even in small concentration are capable of inhibiting the life process of microorganisms. So these antibiotics are produced or derived or produced by living organism, another organism. It may be bacteria, or it may be like fungus, okay, or it may be even synthetic. It may be synthetic, means produced by chemicals, produced by chemical reactions in the laboratory, or it may be like manufactured without the organism. So that is synthetic, or it may be derived, means it may be a part of this microorganism, part of this micro, maybe it may be part like cell wall, cell membrane. It may be part of that, or it may be a chemical produced by this. It may be chemical produced by this microorganism or organism, like it may be a metabolic product of that organism. So all these, it can kill another organism, it can kill the microorganisms. They inhibit the growth of microorganisms. So that's the definition of antibiotics. So why we have to kill another microorganisms? It's like in this actually, it's like we have the words like survival of the fittest. So one organism killing the another organism. So that is antibiosis. That is called antibiosis. That is one organism killing another organism. So that term is that is used here to coin the term antibiotics. Anti means against, bio means life, so against life. So that's how the term came. Now why are we killing other organism? Like, why are we talking about this? That is, this organism which we are trying to kill is microbe, is microorganism which causes disease in us, which causes infections in us. All right, let's go on to that in next slide. Now the antibiotic, a successful antibiotic, or if we want to use that as a medicine, it should be effective against the pathogens. Pathogens are the organisms which cause disease or infections, and it should kill those organisms which cause which should kill those pathogens which cause diseases or infection, and it should do that without producing any toxic effect on the host. The person who are taking the antibiotic, he should not have much of a side effect. It should not produce any toxic effect on the person who is taking antibiotic. So such antibiotics are successful, and those antibiotics should also be stable during isolation. Means they should not, chemicals, they can deform. Not all chemicals are stable. The chemical or the antibiotic which we are talking about, it should be stable during isolation, during production. Isolation in this sense during their production, and storage. While storing them, before we could use them, we store them, right? So while storing, and also it should be stable during its use. While using, it should be in the stable condition, right? Next, let's go into this disease. The disease are caused by microorganisms, right? They are also called microbes. And disease causing microorganisms are virus, bacteria, fungi, protozoa, and worms. So you come across diseases about these organisms, right? These protozoa, they are the ones which cause malaria, and worms. There are worm diseases, like, right, like from roundworm, hookworm. So you're going to come across a chapter on that. We're going to study that in that chapter. So these are all the organisms which cause diseases. In this chapter, we are talking about only about the bacteria. Right. In bacteria, there are two types: gram-positive and gram-negative bacteria. Actually, bacteria, what they do is, in the laboratory, they stain this bacteria. They use the dye and they try to stain this bacteria. Some bacteria, they take the stain, they take the color, they take the dye, whichever dye you are putting it into the test, and when it stains, they call that as a gram-positive bacteria. And there are many examples on this gram-positive bacteria, like streptococci, staphylococci. So again, when the bacteria does not stain, does not take the dye, such bacteria are called gram-negative bacteria. Now, why are they staining and trying to identify the bacteria? There is a, as I told you, you can classify them. You have so many examples here, and you have so many examples here. And also, these gram-positive bacteria, they, suppose we have a person, right? These gram-positive bacteria, they mainly act on the surface of a person, surface of the body, like skin. They act on the skin. These gram-negative bacteria, they mainly act on body inside of the body. They mainly act on inside of the body, especially one on the lungs, and one on the kidney. So they can produce a lung infection and kidney infections. Now there are there are antibiotics which are called narrow spectrum antibiotics and broad spectrum antibiotics. Antibiotics, you can categorize them into narrow spectrum antibiotic and broad spectrum antibiotics. Suppose antibiotics, they act only on gram-positive bacteria, or only on gram-negative bacteria, or only on one type of a microorganism. Such type of antibiotics, they are called as narrow spectrum antibiotics. They are very selective in their action. And antibiotics which cover both gram-positive and gram-negative bacteria, right? Such antibiotics are called as broad spectrum antibiotics. They cover a large number of pathogens. Just they can also even not just gram positive and gram negative bacteria. So they can also cover wide range of microorganisms. So such antibiotics, they are called as a broad spectrum antibiotics. Broad spectrum you can use, just within the bacteria. If within the bacteria, if you are trying to uh, talk about the broad spectrum, that means that those are the antibiotics which cover gram positive and gram negative bacteria. If we are trying to tell that in a broader sense, for concerning all the microorganisms, then those antibiotics, they cover gram positive and gram negative bacteria, including that they also cover other organisms. Right. Next, let's see how these antibiotics, they kill the microorganisms. They kill the other microorganisms. Bacteria. That's the bacteria. Actually, they will have a cell wall. Bacteria, they will have a cell wall. They will have other components there. They will have the DNA and RNAs, and they will have cell wall. Humans, we do not have the cell wall, and because of that cell wall, they are in different shapes. Like they are in a rod shape, they are like round shape. Right? These are called actually cocci. Rod shape, they are called as rods or bacilli. They are called as bacilli, and they can also have like tail-like structure. So that is called as a flagella, and this flagella, it is useful in for them to move from one place to another place. They help them in swimming in the media whichever they are present in. Now these bacteria, they are killed by interfering with the mucopeptide synthesis. These cell wall, they are made with the mucopeptide chemicals, mucopeptide component. Now they can, rupture this mucopeptide. They can even rupture this mucopeptide which is on the cell wall. One thing is, they interrupt with the synthesis. The synthesis of cell wall antibiotics can stop this synthesis of this cell wall, right? Cell wall needs the component called mucopeptide. So this mucopeptide synthesis is interrupted. That's why cell wall is not going to form, and the bacteria is not going to form at all. That's how they stop the growth of the bacteria. They can also interrupt. Suppose you already have the bacteria, and in this cell wall, there will be cross-linking with the chemicals. There will be cross-linking with the chemicals. You have bonds in the chemicals, right? They can act these bonds. They can act these chemical bonds, and they can interrupt this cell cross-linking. These chemicals, they are like cross-linked like this, cross-linked, and the cell wall is formed. And these antibiotics, they can act on this cross-linking, and they can interrupt the cell wall, or they can break that cell wall and kill the bacteria. So they can alter the membrane function of the cell wall. The function of the cell wall is to protect that organism. In here it is bacteria. So they can alter the membrane function. They can disrupt the function of this cell wall, that is the protection, and thereby they can cause harm that bacteria, or they can kill that bacteria. Some antibiotics, they interfere with the protein synthesis. See, this protein synthesis, or they can interfere with the mRNA synthesis. Both these are, even mRNA synthesis, it is required in the protein synthesis. Protein, they produce protein for their growth. So if you interrupt this protein synthesis, then the organism will die, and that's how they kill the antibiotics, they kill these microorganisms. Next, let's go through certain terms. You know what is antibiotic now? So there are terms like antiseptic and disinfectants, especially antiseptic. What is the difference between antiseptic and antibiotics? See, antiseptic, antibiotic. See, antiseptic, they stop the growth of the bacteria. They just stop. Antibiotics, they kill the bacteria. So what is the difference? Like, see, there is think that there is a bacteria. How does it grow? It grows by actually binary fission. It grows by mitosis. You have come across this in your human physiology subject. So they see one bacteria will become two, and these two bacteria will become, again two, and this will become again two. So like this, by mitosis, or by binary fission, they grow. They grow like this, right? Each bacteria will give two more bacteria, just copying. Okay, like this they grow. This antiseptic, they can stop this growth. They can stop the growth. They don't let a new bacteria to form. But antibiotic, what it does is they can kill all these bacteria at once. Antibiotics, they can kill all the bacteria at once. But this antiseptic, it only stops the growth of the bacteria. They don't let the new bacteria to form. They don't let the new bacteria to form. But once the bacteria is already formed, so many bacteria are already formed, they cannot do anything. Antiseptic cannot do anything, but antibiotics, they can kill those. They can kill both, just coming up bacteria and the already formed bacteria. Then what is the difference between, antiseptic and disinfectant? See, both these kill the microorganisms. They kill the microorganisms. Antiseptics, they can be used on us, in the sense they can be used on animals, on our skin. But disinfectants, they are used on inanimate objects. They are used only on objects or surface. Suppose you want to clean something, some object or some surface, suppose rains, or even in the bathroom. So you can use that on the object, on the surfaces. But antiseptics, they can be used on surfaces also. They can be used on our body. Right. Now, there are terms like bacteriostatic and bactericidal. It is the same thing. Bacteriostatic it will just stop the growth of the bacteria. Bactericidal means they completely kill the bacteria. Then there are terms like aerobic and anaerobic bacteria. Aerobic bacteria. Aerobic means air, right? Air means here we refer to oxygen. Aerobic bacteria are those which live in the presence of oxygen. They cannot live without the oxygen. And anaerobic bacteria, they can live without oxygen. They cannot live with oxygen. If oxygen is there, they are going to die. So they live without the oxygen. Then resistance. See, these bacteria, they can show the resistance to the antibiotics. Suppose you have used one antibiotic against one bacteria. Next time you use the same antibiotic on the same bacteria, it may not work. The bacteria might have, undergone some changes depending on its last experience. It can undergo changes genetically inside. It can undergo some changes, and it may not work. The this same antibiotic may not work on the same bacteria. So that is called as resistance. So in that case, you will have to change the antibiotic. You have to go for another antibiotic. Then toxicity. These antibiotics, they can produce harm to our body. So you need to be using antibiotic very carefully. Antibiotics are to be used only when required. They can produce toxicity in our body. So how these antibiotics are produced? How do they manufacture antibiotics? One thing is they can be synthesized, right? They can be synthesized in the laboratory. They can be synthesized. And how are we going to manufacture from bacteria or microorganism? See, in the factories, suppose this is a big tank. This bacteria, the antibiotics. So here they can be prepared from bacteria. Naturally, they can be prepared from bacteria, or they can be prepared from mold. Mold is a type of fungi. They can be prepared by mold. So what they do is they put the nutrients. Ingredients, or it can be even sugar. Okay, and with that they mix with the bacteria or mold. This bacteria or mold, it grows. It shows the growth in this nutrient or the sugar. They grow, and after when this is called actually fermentation. This is called fermentation. Okay, after when this bacteria has grown and produced, they produce antibiotics. This bacteria or mold, they produce this antibiotic from their metabolic process. And this antibiotics, they are extracted. They extract them, means they remove them from this whole growth. They extract them, and then they process this further, and you will get these antibiotics. You can get them in the form of a tablet, or there are antibiotic ointments, right? Ointments or cream, right? They put that into these tablets or capsules, or these ointment or creams. So that's how we use them for treating the diseases or infections. Now let's look into classification on these antibiotics. On the basis of their chemical structure, these antibiotics are classified as follows. First one is beta-lactam antibiotics. So you have come across this beta-lactam ring when we were discussing about the nomenclature. So depending on the looking into the chemical structure, they are divided. So that's how the classification has taken place. The first type is beta-lactam antibiotics. Uh, in this, we are going to come across penicillins and cephalosporins. Penicillins examples: benzyl penicillin, phenoxymethyl penicillin, benzathine penicillin, ampicillin, one with the asterisk mark. We are going to study with the structure. I think I have not put asterisk mark, in the next slides and all. But one with the asterisk mark is with the one we have to study with the structure. So benzyl penicillin, and then we have phenoxymethyl penicillin, benzathine penicillin, ampicillin, cloxacillin, and carbanicillin. Then in cephalosporins we have cephalexin, cephaloridin, cephalothin, and the next class is amino glycoside antibiotics. We have streptomycin, kanamycin, neomycin, gentamicin, vancomycin, and tobramycin. The third one is the polypeptide antibiotics. Examples are bacitracin, colistin, and polymyxin. The next class is polyene antibiotics. Example nystatin, amphotericin, and candicidin. The next is macrolide and lincomycin antibiotics. Examples are erythromycin, lincomycin, and clindamycin. Next is tetracyclines. Example tetracycline, chlortetracycline, and oxytetracycline. Next we have, this is not categorized, so they are miscellaneous antibiotics. Examples are chloramphenicol, griseofulvin, cycloserine, sodium fusidate, novobacin, and rifamycin. So these are the classes of antibiotics, and we are going to study on individual drugs on some of these, right? Some we have to study with structure, right? With structure and with the IUP. Whenever you write the structure, you have to name that structure, give that a chemical name. So we are going to name them according to IUPAC rules. So we have to give the IUPAC or IUPAC names when we write the structure. Okay, that's all for today, and let us study on individual drugs from next class onwards. Thank you very much, and let me know if you have any questions.